Um, we didn't talk about the anticodon part yet, so let's talk about that now. So we said tRNA has a section that can recognize the codon in mRNA. So to give you an example, I have here alanine written out. Alanine, if you remember your codon table, which you'll have access to, you don't need to write that on your cheat sheet, has three forms: GCA, GCU, and GCC. So we said alanine has a specific tRNA. Now, in theory, if you want to recognize GCA, GCU, and GCC, then you should have at least three different tRNAs that have the anticodon to these letters. But that implies that each amino acid has multiple tRNAs, which is too much, and the cell finds a way to actually minimize that. And so one thing we noticed in the alanine is that GC stay the same, but the last letters stay different. So evolution has created a way such that we can actually just make it specific to G and C and let it bind kind of loosely to the last base. And so we call that last base the wobble base. And so if we are designing a tRNA that can recognize alanine, we want a nucleic acid that can recognize the first base. So if you want to recognize G, you want to have a C next to it. If you have a, let's say, let me get rid of this. If you have a G right here, and you want to bond to that, then you can have a C. If you have a C, you want to have a G. And for the last base, you want it to bind to multiple ones. And so the way to solve that is through this example right here, where you have a base called inosine. And inosine is special in that it can bind to both A, C, and U just with one structure. And what you'll notice here is that the part of inosine that binds doesn't change. It's still these two parts. However, it just binds to different parts of the partner. And so instead of having a specific one for each of these nucleic acids, you can just have C, G, and then I. And that CGI structure on the tRNA can recognize GCA, GCU, and GCC. And so this allows for redundancy. And this way, the cell can only have one tRNA for alanine that can recognize all of the codons for alanine. And so it's a very common question to ask about the wobble base. And so if ever there's a question about how can a single tRNA recognize multiple codons, they probably utilize a wobble base by having a specific nucleic acid that can recognize multiple ones. And lastly, generally when we do translation, the first amino acid is usually N-formal methionine. So usually it'll recognize methionine tRNA, but this special methionine has a formal group attached to the amino terminus. Yeah. Before I end, I kind of want to give a small introduction to translation, and we'll continue next week after the exam. So translation is done by the ribosome, as you guys probably remember from intro bio. So one thing to remember is that the ribosome has two parts to it that are important. One is the protein part, which in here you'll see in light purple. And then the RNA is the gray and the blue. And what you should appreciate is that in the structure right here, the part that is closest to the RNA the mRNA that is the RNA molecules and not the protein. And so when you think of the ribosome, it's basically a protein shell, and the factory inside it is actually the rRNA, the ribosomal RNA. So that's one thing to emphasize on when you watch Yafi's lectures in the future. The other thing is that the ribosome has a large subunit and a small subunit. And so normally ribosomes are named after their sedimentation. So the unit for that is Spedberg, so how far they sediment. The larger the number, the heavier is the general term. So when you have a prokaryotic ribosome, there's a large subunit that is called 50S because its sedimentation or Spedberg is 50, and then a small subunit 30S. And together, we call them the 70S ribosome. And I know the arithmetic doesn't add, but if you actually combine these two subunits, its sedimentation is 70. So just remember why it doesn't add mathematically. Eukaryotics have a similar one, but it's a little different. Instead of 30 plus 50 equals 70, they have 40 plus 60 equals 80. And as I said earlier, most of the work in translation is governed by the RNA. So this really supports the idea that RNA is the first living molecule that can both replicate and act as an enzyme. Moving on. I'm not going to cover this because it's not yet covered and hopefully you haven't even watched it yet. But you're going to learn the steps of translation. And it's going to be kind of like this in terms of recipe of which molecules and which proteins function. What I want you to kind of remember is that focus on the costs. So he's going to start saying this step requires 1 GTP or this step requires 1 ATP. Definitely focus on that. It's a very, very common question to ask you. How much GTP or how much ATP do I need to undergo translation or to undergo transcription? Or in this case, it's only translation. So make sure when you watch those videos to put special attention because this throws people off all the time. And it's a simple math if you know the rules. So make sure you focus on the cost. Anywho, I hope that was a good cover of the material so far. This is a new format, and all recitations are basically just this now. And so I would love to have your input. Go to this link or go to the link in the Stellar page to be able to kind of give us feedback. I am contemplating on having a Zoom version potentially. That way I can answer questions in real time. But if there's any suggestions or if I can do anything to improve the recitation, then let us know. If you also have questions on the material or the course in general, feel free to email us or put it on the survey. Other than that, I'll see you guys next week.